As I was saying, somebody must have scheduled me before lunch because I'm going to be talking about food webs. Food webs, quite simply, are the lion eats the zebra eats the grass. Pretty simple. Of course, there are more species than just 3 in an ecosystem. You can have a whole variety of plant species, herbivores, carnivores, all the parasites, all the other things I'm not showing in here. It gets very complicated very quickly. Here's another picture of a food web. This is from an ecosystem in South Africa, a marine system. And you see these are actually aggregated nodes in this network. So it's actually much more complex than this. And so this leads you to think, well, where does all this complexity come from? And how does it all work? And what does it all mean? But today I'm going to focus on a slightly simpler question: is what meaningful patterns do we find in observed networks of feeding links? And hopefully this much simpler question can start to inform us as to how to answer that deeper, more complex question. So first, how are we going to do this? The food web data I'm talking about today is simple. It's just a binary matrix of who's eating whom in the ecosystem. So column 9, row 3 being filled in means that species 9 eats species 3. And the approach for finding patterns in this data is first to formulate some hypotheses about how the food web is structured, what's important for structuring it. Encoding these hypotheses as Bayesian probabilistic models. And then you fit those models, and I won't talk too much about how to do that, and then you compare the fit of those models to data. And finally, interpret model parameters in order to hopefully learn something about the ecosystem that you're studying. So, probabilistic food web models, pretty simple. For every pair of species in the food web A and B, we want to be able to define what's the probability of A eating B. That's going to be some function of the characteristics of these two species. The characteristics may actually be empirical traits, the size of the jaw, the speed of the animal, the type of chemistry in the plants that you may have measured. Or if you don't have any of that, you may be able to describe this just in terms of some kind of abstract inferred trait space, which are going to be the parameters of the model. And then to get the probability of the entire food web, given your model and your parameters, you just multiply all the pairs of possible links in your food web. So luckily we got an introduction to Bayesian statistics earlier, so I'm going to do this in a Bayesian framework where you have a prior distribution over your parameters, you observe your data, and that gives you a posterior probability distribution over the parameters describing your model. So what are the patterns that we're looking for in these food webs? One very simple, almost null model is to say that our ecosystem is going to be made up of guilds of species that have statistically similar patterns of eating and being eaten by other groups of species. And so here we have 3 distinct plant groups, a couple distinct herbivore groups, and a carnivore group. You'll notice in the middle, I put the elephant with this little critter called the hyrax, which may strike you as odd, but you'll see that it actually works out like that. And so if you're to look at the adjacency matrix of the network, this is going to look something like this. You have diagonal and mostly off-diagonal blocks, which is different from normal network modularity, where you're dealing primarily with on-diagonal blocks. And if you want to actually formulate this as a probabilistic model, you need to infer both which species goes in which group, and also parameters controlling the linkages between the groups. What's the probability that a species in one group is eating a species in another group? Another pattern is to bring an actual biological idea into this model, which is to say, all right, imagine that we can't measure all the different traits that control who's eating whom in the food web, but let's imagine that there's some simple one-dimensional projection of all of those traits. So we're going to line up all our species on this line. Each species gets a dot on the line, and that determines who eats that species. In addition, each species is going to get an interval along the line determining who it eats. And so we have here this dark blue species has an interval there, which spans 4 species. meaning that that single species eats those 4 species. Everybody gets an interval, everybody gets a dot, and those are going to be parameters we're going to want to infer. And so this pattern manifests itself in the adjacency matrix like this. We have contiguous blocks of species in the columns, assuming we've ordered our network in the right way. One dimension may not be enough. If you have 2 species with overlapping feeding niches like this, you can always do it with 2 species, but if you add a third that covers all of these prey items, except for these 2 in the middle, there's no way to order these so that everybody has a contiguous feeding niche. So you move those out of the way into the second dimension, and now these 2 species can have the same overlapping feeding niches. The third species can now be restricted in the second dimension, and it all works out. And so that's going to manifest itself in our food web adjacency matrix with gaps if we try to order everything in 1 dimension. So that wasn't a probabilistic model. I just said if you eat within an interval, or if you have a particular interval, you eat everyone within it. If you want to make it probabilistic, you turn it into a fuzzy interval so that if you're out at the edges, you're less likely to eat something. So here, right at the center of the circle where it's white, you say the probability of this particular predator eating dots in the middle of its feeding range is 1, out at the edges it drops to 0. And so you're going to want to infer both the locations of these species in feeding niche space in 1 or 2 dimensions, and also where they eat within that space. Finally, you can combine these 2 patterns together and say, we're going to look at some sort of high-level decomposition of our food web into functional groups, but we're also going to put these things into a niche space. And the way we do this is by using a Bayesian hierarchical modeling framework, where we organize the species into groups, and within the groups, each species is going to have its parameters drawn from a common prior distribution. And then each of those parameters controlling those distributions will come from a hyperprior, and then we'll infer our posterior distribution over both the groupings and the niche space parameters. All right, so back to some biology and some results from the Serengeti. Serengeti National Park is in Tanzania. I've never been there. I'm hopefully going to go in late November, early December to do a safari and a conference. And so take anything I say about the biology with a grain of salt. I've never actually seen it. In this data set, we have 9 mammalian carnivore species, 23 mammalian herbivore species, and 129 plant species, resolved down to the species, in some cases, the genus level. And first, just a high-level view. If you just do a functional group analysis, you get a series of different plant groups which seem to be associated with particular habitats in the food web. And you get also a decomposition into distinct different groups of herbivores and carnivores. I'm not going to dwell on this one too much, because this gets refined. Because if you look at the ranking of these different models, we find that the best model, fairly significantly, is if you take a 2-dimensional niche space and combine it with this hierarchical group structure. So we're decomposing into functional groups, and we're also placing these species into a 2-dimensional niche space. In second place, the 1-dimensional model. In third place, just functional groups, and then it drops from there. So this 2D niche space with groups is statistically the best supported description of our food web, of these alternative models. So when you fit this model, you get a distribution again of these parameters. So one of the parameters is which group are you in. This is a similarity matrix for the herbivores. The color indicates what's the probability of 2 species being in the same group. You can take this similarity matrix and cluster it and get sort of a summary of this posterior distribution of groupings. And the summary says we basically have 2 big statistically similar groups of herbivores that are similarly positioned in niche space. What's interesting, if you look at these groupings and look at how these species are related to each other in terms of evolution, all of the Bovidae, the family that contains cows and all of these grazers, get stuck in the same group with the exception of a couple species, and the exceptions are in fact the ones that are most weakly put into the group on the top. And you can test for this, and this is statistically significant. So we're seeing a reflection of evolution in this food web, which isn't surprising because closely related species have similar traits. They have similar types of stomachs. They have similar jaws, et cetera. And so this sort of evolutionary relationship we can think of as a proxy for the biological traits of the species. And I pointed out earlier that the hyrax and the elephant, they actually make sense to be put together. If you look at the clustering of these groupings, they get put right next to each other. If you look at the evolutionary tree, in fact, the hyrax, which is this little thing, just a few kilograms, is the most closely related living species to the elephant, which seems shocking. But if you actually look at what they eat, they share more of their diet than these other species who have different kinds of stomachs, different kinds of behaviors, even though these two species look totally different. So it's interesting. The plant groups, you have a similar pattern. You can decompose it into large functional groups, and you can test for evolutionary relationships here, and species that are more closely related evolutionarily are more clustered. So we get a schematic bird's eye view that says, all right, we've got some big functional groups of plants, herbivores, and carnivores. Now let's look a little deeper. We can look at the niche space. This is the same picture I showed you before is for the rock hyrax, and each dot is one of the plant species. It's blue if in the data set the hyrax actually eats that species. It's red if the hyrax doesn't eat it. So you see, the model isn't capturing it perfectly. You'd want all of the blue ones to be right toward the center, all of the red ones to be out toward the edge. But you have to balance all of the different species demands within this two-dimensional niche space. And so if you smash all of the different species feeding niches, all of these Gaussian or slightly non-Gaussian blurs in this feeding niche space, you end up with a map that looks like this. So each circle indicates the diet, the 50% probability boundary of the diet of each species. And the gray background indicates all of those different probabilities added up. So at each point in this diagram, you get the expected number of predators you're going to have if you're at that point. And you can see that the blue ones are drawn from the same prior distribution, and the yellow ones in the middle are drawn from the same prior distribution because they're in the same group. And in here, you can also detect evolutionary relationships. If you're closer together in this inferred niche space, you're going to tend to be more closely related to each other. Here's the same diagram for the herbivores and the carnivores. See, the carnivores get sucked out to the edge. Way in the upper right, we have lion, hyena, and the cheetah, who don't get eaten by anybody else, but they eat some of the other carnivores. So they're contained within the feeding niche space of those larger carnivores. All right. So conclusions: a two-dimensional niche space, when combined with groups, is the best model for describing this food web of these different alternative hypotheses. A 2D niche space, interestingly, it's statistically superior to one dimension only if you use this dimension reduction techniques of grouping species together. So that clustering is actually necessary. And also this food web structure reflects evolutionary relationships among species, which you can think of as a proxy for biological traits. And how am I doing? Am I out of time? 15 seconds, all right. So I'll talk for 15 seconds about computation. All these parameters are being inferred with Markov chain Monte Carlo method. We have a series of configurations of parameters. Between each of these, we propose a change to the parameters, accept, reject. And there are 3 different methods for doing this: Gibbs sampling, Metropolis Hastings, and also this thing called differential evolution MCMC, which takes into account the correlation structure of the parameters without actually tracking the full correlation matrix. And it does this by keeping a history of the parameters. And so if you basically take 2 random samples from your history, the differences between those historical samples in your chain are going to be correlated in the same way your parameters are. So that can improve convergence quite a bit. This will converge to your posterior distribution eventually. I had to use one cheesy effect. So you're going to have a whole bunch of chains, and the chains, as you go farther down, to use a thermodynamic analogy, are heated, so they explore the distribution more freely. And then if you get a better configuration for your parameters, that better configuration can trickle back to the cold chain, and that helps you explore parameter space more effectively. One more. And this is put together in a general purpose framework for inferring Bayesian probabilistic models. It still has some warts, but the code is on GitHub, and I'm hoping that'll be more generally useful in the future. Skip that. So thank you. I'll take any questions. My advisor is Mercedes Pascual. The Serengeti expert who took most of these photos is Andy Dobson. Other collaborators are Stefano, Trevor, and Mike. And of course, thank you to Krell and the DOE CSGF for providing me the opportunity to work on this stuff. Thank you very much.